In our original experiments, when we monitored the conjugation of ubiquitin to a single specific proteolytic substrate, we found that ubiquitin conjugates are migrating all the way to the very top of the gel to a very high molecular mass region. This told us that either the substrate is conjugated by multiple single ubiquitin moieties attached to different internal amino acids, or that a polyubiquitin chain anchored to a single amino acid must be generated in order to target this substrate for degradation by the proteasome. In a very elegant experiment carried out by Avram Hershko, he blocked all the internal lysins of ubiquitin chemically and found that this modification blocked the degradation of the substrate altogether. And his conclusion was that we need a polyubiquitin chain. Actually, he coined the term in order to target a substrate for degradation by the proteasome. And this has become the hallmark of the ubiquitin system. A polyubiquitin chain is a pre-requirement for targeting a proteolytic substrate for degradation by the 26S proteasome. Meanwhile, the structure of the proteasome has started to emerge. Initially, the first subunit that binds ubiquitin was discovered, then the second one, then the third one. But however structural biologists looked at the proteasome, it was not clear why do we need a polyubiquitin chain made of tenths or several tenths of moieties of ubiquitin, and even more, maybe close to 100 moieties of ubiquitin, in order to bind the substrate to the proteasome. At the maximum, the distance was of 4, 5, 6, 7 ubiquitin moieties, but not more. Then why multiple ubiquitin moieties linked to one another in a chain? One theory was that we build this chain in order to combat deubiquitinating enzymes, and then the chain is being trimmed, edited to the right size. But the theory has never been proven. We decided to test this hypothesis from the very beginning, and we developed several tools for that. And initially we found that a single ubiquitin molecule can target proteins of up to 150 amino acids for degradation, which made some sense that you need just a single ubiquitin moiety in order to bind short peptides, but in order to bind longer proteins, you need probably a more tight linkage between the ubiquitin moiety and the proteasome, so the substrate, the ubiquitinated substrate, will not tether and dissociate from the proteasome and will render proteolysis processive. Then we discovered that another substrate, which is a longer substrate, the P105 precursor of NF-kappa B, requires multiple monoubiquitinations in order to be processed in a limited manner to generate the active P50 subunit of the active NF-kappa B transcription factor. These two experiments with the short proteins and with NF-kappa B told us that there is no pre-requirement by the proteasome to bind a polyubiquitin chain. And the proteasome can bind either a single ubiquitin moiety or multiple ubiquitin moieties scattered along the substrate. These two experiments prompted us to do a systematic search for all proteins that can be targeted by either monoubiquitination or multiple single monoubiquitinations. And what we did, we expressed in cell, we shut down the native ubiquitin genes, and we expressed in either yeast cells or mammalian cells a non-polymerizable ubiquitin, a ubiquitin in which all the internalizing residues were replaced by arginines. And surprisingly, or as a matter of fact in retrospect, not surprisingly, we discovered that a large fraction of both the yeast and the mammalian proteomes can be targeted by the proteasome following single ubiquitination or multiple monoubiquitinations. As a matter of fact, the method didn't allow us to distinguish between the two because we didn't touch the internal lysins to which, in the substrates to which ubiquitin is attached. But nevertheless, the conclusion was firm. Single ubiquitination or multiple monoubiquitinations can target the multiple substrate for degradation by the proteasome. But the system didn't allow us to distinguish between either a single monoubiquitination or single or multiple single monoubiquitinations to polyubiquitination. And the question is whether polyubiquitination is at all necessary. My belief is that not, that we don't need a long polyubiquitin chain, and different substrates may vary among themselves in the length of the polyubiquitin chain. We probably need 5, 6, 7, 10, maybe 15 ubiquitin moieties in a chain in order to target those proteins that do need a chain. And the initial hallmark that was a true long polyubiquitin chain was an artifact of the in vitro system that we used either in vivo in cells when we are using proteasome inhibitors, or in vitro when we are using both proteasome inhibitors and the deubiquitinating enzyme, the enzymes inhibitors, in order to trap what we thought until recently, and probably mistakenly, as the signal for degradation, which was polyubiquitin chain, but now I believe it's an polyubiquitin chain or single moiety or several single moieties of ubiquitin. So time changes the signal, and we now, I think, can define it more precisely.